plant breeding. Uh, Mainly, these are the main characters. The extra chromosomes found in the plants, insects, some of the vertebrates, not part of the standard the chromosome set, often the smaller and non-essential, and may not the follow the Mendelian inheritance. What are the functions of the beta chromosomes? Can influence the gene regulation, fertility, and adaptation. Sometimes act as the selfish genetic elements sustaining in the population despite no clear advantages. Next, in the holocentric chromosomes. These are the characteristics of the holocentric chromosomes that are found in the certain insects like Lepidoptera, butterflies, nematodes, and some plants. A lack of a single centromere, the spindle fibers attached along with the entire length. Chromosome breakage does not lead to loss of the genetic information. What are the functions of the holocentric chromosomes? They provide the resistance of the damage and breakage, facilitate unusually modes of the inertancy and chromosome evolution. Microchromosomes, these are the special type of the chromosomes. These are the characteristics of very small chromosomes found in the birds, reptiles, amphibians, and some fishes. Contain the highly condensed genetic material. In birds, they make up in the significant the portion of the genome. What are the functions of the microchromosomes? Contribute to the high recombination rate, leading to genetic diversity, play a role in sex determination and evolutionary adaptation. Next one, ring chromosomes. These are all the special types of the chromosomes. What are the main characteristics found when the chromosome losses? It's the telomeres and circumambulation can occur spontaneously or due to the DNA damage associated with the genetic disorders such as the ring chromosome, 14 syndrome, and ring chromosome X and turnover syndrome variant. Main functions of the ring chromosomes mainly the cause the genetic instability leading to the developmental disorders. Using the research on the chromosome behavior and the abnormalities. Isochromosomes. Characteristics of the isochromosomes is the abnormal division of the chromosomes leading to two identical arms. Common in certain genetic disorders like the turnover syndromes and isochromosome XQ and the some cancers. Okay, isochromosome. What are the functions of the isochromosome? Can lead to the genetic imbalances affecting of development genetic imbalance in the study of the medical genetics and understanding the chromosome mutation. Special types of the chromosome plays the significant role in genetics, evolution, and the disease. They help the researchers in understanding genes expression, chromosomal behavior, and hereditary disorders. While some of the adaptive, other cause of the abnormality linked chromosomes, these are the chromosomes, special types of the chromosome. Next topic is the cell cycle and apoptosis. Okay, the cell cycle. The cell cycle is a series of the order events that lead to the cell division and duplication. It consists of interphase including the G1, S phase synthesis, G2 phase, and mitotic phase, M phase, followed by the cytokinesis. Phases of the cell cycle: interphase, preparation for the division; interphase, G1 phase, the first gap phase, cell grows in size and synthesis of the proteins and the organelles. The cells checks for DNA damage before proceeding to the S phase, synthesis phase. And the next one is the S phase, synthesis phase. Mainly in synthesis phase, DNA replication occurs producing the two identical sister chromatids. Histones and other proteins are the synthesized. Due to the second gap phase, cell continues to grow and the prepare for the mitosis and DNA check for the errors and the repair if necessary. These are the interphase G1, S phase, G2 phases. Next one, M phase, mitosis and cytokinesis. Mitosis, mitosis is the nuclear division. Mitosis also they are containing the prophase, the metaphase, anaphase, telophase. Prophase, chromosomes condensed in the spindle fibers are formed in this phase, prophase. Metaphase, metaphase in chromosomes aligned at the cell equator. Anaphase, sister chromatids separates and move to the opposite poles. Telophase, nuclear envelope reforms around the separated chromosomes. Cytokinesis and cytoplasmic division. The cytoplasm divides the forming into identical daughter cells. What is the regulation of the cell cycle? The cell cycle is the tightly controlled by the checkpoints and regulatory proteins to prevent the uncontrolled growth cancer. Okay, somatic dividing cells undergo the process termed as the cell cycle. The cell cycle is ordered the serious events leading to the cell replication. The goal of the cell cycle is to produce in multicellular organisms, most differentiated cells dies. already in okay, what are the regulations of the cell cycle? The cell cycle is controlled by and regulatory proteins to prevent the uncontrolled growth cancer. What is the checkpoint? And G1 checkpoint, the restriction point is also called as the restriction point. Checkpoints checks for the DNA damage before entering the space, regulated by the P53 tumor suppressor protein, the P53. G2 checkpoint is the ensures the DNA is fully replicated and undamaged before the mitosis. M checkpoint, spindle checkpoint ensures the all chromosomes are the attached to spindle fibers before anaphase. Regulatory proteins, check cyclins and the cyclodependent kinase CDKs. These are the regulator protein. Control the progression of the cell cycle. Different cyclins, the activity of CDKs at different phases. Tumor suppressor, especially P53 RB protein, is the both are the tumor suppressors. Alter of the cell cycle errors are the detected, preventing the cancer development. Apoptosis. Apoptosis means the cell death or the mainly mechanism of the cell control in all eukaryote the cells universally control the mechanism regulates the events of the entry into G1, S, G2 and M phases. Already we discussed the G1 phase when we are the stimulate the divide and G1 cyclin CD complex are the expressed in the first. The initiation starts with the rising levels of the cyclin D in the cytoplasm. Cyclin D binds to CDK4 and enters the nucleus and the signals of the cell prepare of the chromosome for replication. S phase is the rising of the levels of the cyclins in E and A type of the bounded CDK2 are called special type of the phase promoting factor SPF enter into the nucleus and prepare the cell duplicate its DNA. DNA replication continues. The cyclin E is the destroyed. T2 phase is the level of the cyclins B and A type of the begins rising during the phase. M phase metastasis initiated with the binding of the cyclins B type with the CDK1 causing assembly of the mitotic spindles, breakdown of the nuclear envelope, and convention of the chromosomes. What are the steps are involved in the cell cycle? The first one is the DNA pre-replication complex assembly at the origin. Next one, G cyclin CDK inactivated the CDH1. Next phase is the G1 cyclin CDK activated the expression of in this phase cyclin CDK components. G1 cyclin CDK phospholipids and this phase is the inhibitor. SCF already protosome and degrades the phosphorylated the S-phase. Next one, the cyclin CDK inhibitor. S-phase cyclin CDK activates the pre-replication complexes. Mitotic cyclin CDK activates the mitotic events. APCDC20 and proteasome degrades of the securin. APCD and the proteasome degrades the mitotic cycle. These are the all cycles are the mainly um, cell cycle. Those are the stage. So already checkpoints is over. And the reasons for the programmed cell death. What is the meaning of the programmed cell death? Mainly, programmed cell death is the apoptosis genetically control the process, eliminates the damage. Apoptosis means programmed cell. Apoptosis is genetically controlled process that eliminates the damaged, infected, or unnecessary cells. It is essentially for the development, immunofunctions, and the cancer prevention. What are the characteristics? Cell shrinkage, the loss of the membranes, asymmetry, chromatin condension, and DNA fragmentation, formation and apoptotic bodies, which are the amplified by the phagocytes, no inflammation, unlike necrosis. What are the molecular mechanism of apoptosis? Mainly, apoptosis occur through the two major pathways, intrinsic pathway, mitochondrial pathway is also called as the mitochondrial pathway. Intrinsic pathway is also called as the mitochondrial pathway, mainly triggered by DNA damage, oxidative stress, or lack of survival signals. The BCL2 family regulates the mitochondrial membrane permeability. Cytochrome C is released from the mitochondria, activating the capsase 9, leading to the cell death. Next one is the extrinsic pathway, death receptor pathway. Extrinsic is also called as death receptor pathway. Triggered by the external signals, fast ligand binding the fast receptor, activate the caspase 8, which is triggers downstream caspase leading to the apoptosis. Next one, execution phase is the final stage. Next one, next phase is the excretion phase away. Caspase 3 and caspase T degrade the cellular components. The cell is dismounted into the apoptotic bodies and removed by the phagocytes. What are the difference between the apoptosis and necrosis? What are the causes of the apoptosis? Main cause is the genetic program. Necrosis injure the infection. Cell size shrinks, mainly shrinks in the cells of the in necrosis, and apoptosis is mainly shrinking of the apoptosis cells. DNA damage fragmented in apoptosis and random degradation in the necrosis. Inflammation, there is no inflammation in the apoptosis. Inflammation is the necrosis, is the S. Outcome of the phagocytosis is the apoptosis and cell burst into the necrosis. Importance of the Importance of cell cycle and apoptosis. Growth and development ensures the proper formation and tissues and the organs. The importance of the cell cycle and apoptosis. Growth and development ensures the proper information, tissues, and the organs. A tissue homeostasis balances cell division and the cell death. Prevention of the cancer. Apoptosis eliminates the abnormal cells before the can become the cancerous. Immune system functions, removes the infected and damaged cells, prevent the disease spread. Genes are the controlling of the apoptosis, regulating of the apoptosis. Next one, cell survival signals, signals of the apoptosis. Okay, mainly apoptosis results from the action and the special category of the protease called capsid, which is exit the cells and inactive processes by the proper triggering, and the capsid leads the amplification of the chain of phosphase cascade, which is the finally results the cell death. The mechanism by cascade activation is the further regulated differently in the different organisms. Death of the certain cells during the cell culture also results in the part of due to the apoptosis, involving the expression of the certain genes and addition to the signals with the functions opposite directions to make the cell survive. This conduct the studies apoptosis in the PCD program cell death the healthcare therapy. Next one is the DNA. DNA is the basic unity of the identity. Initially, gene was the regarded the participant and was the correlated with the chromosome structure. During the last 30 years, the gene were the regarded the beds and the string of the were mapped in the diverse organism. Recent advance in the field of the molecular. What are the important features of the DNA? Qualitative estimation of the DNA carried the various organisms, the following some of the features. A genome is a single copy of the genetic information of the cell. The coded information within the genome is used to control the synthesis of the proteins. The sequence of genomes and determines the sequence of amino acids in the proteins. Gene is a segment of DNA molecule. Alleles are the different forms of the same gene, and the amount of the base composition of DNA is the constant for the organism, but is the various different species. DNA is the called of the deoxyribonucleic acid. DNA is the polynucleotide, mainly is the made of the many units of the polynucleotides. DNA is the macromolecules due to the high molecular weight. DNA is the hereditary substances. DNA is the life molecule. The chemistry of DNA is the proteins which is the linked with the DNA chromosomes known as the basic proteins or histones, which are the rich in the amino acids, lysine and arginine, and have the net positive charge. On hydrolysis of the molecule yield nucleotides, each nucleotide is the composed of 4 nitrogen bases, pentose sugars, and deoxyribose and the phosphate. The nitrogenous bases are the 2 pyrimidines, the cysteine and the thymine pyrimidines, and 2 pyrimidines, adenine and guanine, AGs. Okay. In a molecules, 1 pyrimidine base pairs with the purine base, A double bond T and G triple bond C. In DNA, number of molecules guanine is equal to number of molecules of the cytosine. Okay, the cytosine. The number of molecules of adenine is always equal to the thymine. Okay, where the A equal to T and G equal to C. Actually, A plus C equal to G plus T. The base of the pyrimids are attached to the deoxyribose with the carbon atom, phosphocarbon atom. The phosphate is attached to the 5 carbon atom. The phosphate covers up the bounded the 5 carbon atom. One sugar is the covalently bonded to the three carbon to such the DNA strands that twisted around each of the special stages. Deoxyribonucleic acids, DNA is the double-stranded helical molecule that carries the genetic instructions for the growth and development, functioning and reproduction of the all living and the many viruses. Double helix structure discovered in the James Watson and the Francis compose the nucleic ribose sugar phosphate nitrogen bases, the guanine base. Base pairing rule. A pairs with the T two hydrogen bonds. C pairs with the G adenine pair with the thymine. A double bond T and C pairs with the G is the three hydrogen bonds. Structures of the DNA. The two anti-parallel strands in opposite directions, 5 prime to 3 prime and 3 prime to 5 prime. The strands are held together by the hydrogen bonds between the complementary base pairs. DNA is twisted into the right-handed helix with the approximately 10 base pairs per turn. Components of the DNA, nucleotides, the building blocks of the DNA, the sugar phosphate backbone provide the structural stability, nitrogen bases, storage the genetic information. What are the functions of the DNA? Genetic information storage. DNA stores the instruction of the for making proteins. Replication of DNA is copied before the cell division to the ensure genetic continuity. Protein synthesis, DNA codes for the RNA which is used for the produce the proteins. Mutation and the evolution changes in DNA sequence drive evolution. DNA replication is a semi-conservative process, meaning each new DNA molecule has one original strand and one newly synthesized Central dogma of molecular biology, the following genetic information, DNA to RNA, to protein, DNA to RNA formation is also called as the transcription, occur in nucleus into the messenger RNA mRNA. Translation translate to the amino acid forming the proteins. Types of the DNA. Nuclear DNA is the location is the nucleus. The mitochondrial DNA location is the mitochondria. Inheritancy from the both parents inheritancy, and only the from mother is the mitochondrial DNA. Structure is the nuclear DNA is the linear and mitochondrial DNA is the circular. Different forms of the DNA, aDNA, compact found in the dry conditions. And bDNA is common from the in all the cells. When JDN is the left-hand helix, may is the play the role in the gene regulation. Mutations in DNA, mutations are changes in the DNA sequence which can be Okay, importance of the DNA, essential for the heredity and evolution. Is DNA fingerprinting in the forensic science, key to genetic engineering and the CRISPR gene editing, mainly genetic recombination in bacteria, transformation and conjugation. And next one is the transduction. Mainly transformation DNA is the discovered in the Greek transformation. Conjugation is the 1940s Lederberg and Totem discovered in the E. coli. Okay, conjugation. And next one is the transduction. Mainly is the transfer of the genetic material from one bacterial cell to another. Means the viruses is known as the transduction. These are also discovered by the Ginder and Ledenberg in 1952. Mainly these are seen to the Salmonella, P22 and Salmonella. These are the DNA. Main functional aspects of the DNA: genetic information storage. DNA encodes the instruction for the synthesis of proteins and the functional RNA. The sequence of bases determines an organism traits. DNA replication, semi conservation conservative model ensures the genetic continuity during the cell division. Key enzymes are involved in the DNA replication: helicase, unwinded DNA, DNA polymerase, synthesis for the new strands, ligase, joins the Upajaki fragments and the plugging of the strands. Central rogama, transcription DNA to RNA, translation RNA to protein. Evolutionary aspect of the DNA, a genetic variation in the mutations. Mainly mutations in DNA create the genetic diversity and the drive evolution. Types of the mutation: point mutation, single base change, frame shift mutation, insertion and deletion, chromosome mutation, large scale changes. Horizontal gene transfer, transfer of DNA between organisms outside of the reproduction. Importance of in antibiotic resistance and the medicinal evolution, horizontal gene transfer, HGT. Molecular phylogenetics, DNA sequence is used to the study and evolutionary relationship, mitochondrial DNA. Mitochondria is the widely used in the tracing of the ancestry. Technological aspects of the DNA, mainly using the DNA fingerprinting, used in the forensic science and the pattern testing, based on the variations and short random repeats in STRs, genetic engineering and the recombinant DNA technology, modifying the DNA for medical and agriculture and industrial purposes. Techniques including the CRISPR-Cas gene editing, gene cloning, producing the identical DNA copies, transgenic organisms, GMOs, DNA sequencing, determine the precise order of the nucleotides in DNA. Next generation sequences of the as a revolutionize the genomics, medical and clinical aspects of the DNA, DNA diseases, genetic disorders caused by the mutations, sickle cell anemia, cystic fibrosis, and cancer genetics, mutations in the oncogenesis or tumor suppressor genes, P53 mutation in the cancer, epigenetics, environmental factors affecting the gene expression without the changing the DNA sequences, gene therapy, correcting and defective genes in the genetic disorders, used for the disease like SCID, severe combined immunodeficiency, pragmacogenomics, the studying of the genetic variation affecting of the drug response, helps the personalized medicine, DNA and the synthesis biology. Artificial DNA synthesis. Scientists can the synthesis DNA sequence for the research and the medicine purpose, used in the developing vaccines and the synthetic life forms, storage and the biological computing for the long-term data arching, cloning, reproductive cloning, creating and genetically conclusion of the DNA is the multifaceted molecules with the roles of the identity, evolution, technology, and synthetic biology. Understanding DNA is the transfer many fields in the forensic to cancer treatment. Advanced like the CRISPR gene editing potential of DNA science. These are the DNA. Mainly use the DNA structure, DNA replication, central dogma mutation, genetic disorders, next one DNA sequencing and the genetic engineering, DNA in medicines, mainly this is for the gene therapy, cancer genetics, and oncogenesis, evolutionary aspects, molecular phylogenetics, and the mitochondrial DNA, DNA in biotechnology, the fingerprinting, polysonic applications, the cloning, the all aspects also mechanisms for the DNA.